Hello everyone. My name is Rajat and I'm a PhD graduate from Purdue University. Uh, Today I'm going to talk about a very interesting topic, which is one of the recent advancement in the field of artificial intelligence and large language models. And the main topic and the area which we are going to look at today is proteins. So let's first understand a little bit about proteins. So proteins are basically the building blocks of our bodies, and every single protein uh, is being made to target a specific function, and that function can be, let's say, digesting food. It can be movement of your muscles, it can be fighting diseases. So there are a lot of functions which our bodies do, and every single function can be mapped to a protein or a sequence of proteins. Uh, and proteins are basically made up of a bunch of amino acids, and these amino acids, there are 20 different amino acids, and every single protein has a unique combination and a set of amino acids which is called as the sequence of a protein. So, in the latter half of my PhD, I was actually working on a very specific type of proteins which are known as monoclonal antibodies. This might be new to some of you. So, for humans who cannot develop their own antibodies or their own proteins to resist diseases, people inject these monoclonal antibodies or MABs, uh, which are artificially injected in our bodies to fight these diseases. And this was relevant especially during COVID 19. So, monoclonal antibodies have a very specific shape, which is called as a Y shaped structure. And within this structure, there are these bunch of amino acids which are residing within each domain. So there are three broad areas within which the amino acids reside. And so basically, the point I'm trying to make here is that every protein has got a sequence, which is a sequence of amino acids. It has got a structure, which is a three-dimensional structure of the protein, which is pretty much unique to that particular protein. As you can see here, it has a Y-shaped structure, and then every protein has a specific function as well. So the function of these monoclonal antibodies would be to fight off the infection which is posed by an invading virus uh, in our bodies. And so every single protein has a unique set of sequence, structure, and function. And these proteins have actually evolved through millions of years, or even billion of years. And the reason why evolution has taken so much time is because of mutation. So maybe initially, before we get to these antibodies, initially we had only the first domain, and then after a few centuries we had two domains. Then slowly we started to learn about connecting these in the form of a Y shaped, and then finally we get the antibody. And this whole process, this whole sequence of steps, it usually takes a very long time. It's the process of evolution, that every single mutation is being studied, and whichever mutation survives, that mutation is being carried on in the future generations. So this takes a very long time in general. And any new protein that, let's say, scientists go out and they see a new protein, it is very rare to synthesize a new sequence and a new structure in the lab. Now, another problem which is quite different from this is that how can you predict the structure given the sequence? It is not very straightforward. Google released a model called AlphaFold, which was a big revolution. It used AI to kind of predict the structure from the sequence. Now, the question that we are trying to ask here is that, can we basically mimic evolution? can we mimic evolution? And it looks like a pretty strange problem even when I speak about it. And the reason it looks strange is because evolution is something which takes a lot of time. And how can we, and what do I even mean by this question? So what I mean by is that, can we basically skip all these millions of years of evolution, and can we predict newer and newer proteins just by understanding data? So the main concept is that we take a huge repository of sequences, then structures and functions of all the different proteins that we know in the world, which exist in the world. We collect a huge data, we pass it through a large language model, and then we develop the capability to predict new structures and new functions of new proteins. And this problem inherently sounds okay. Like, does it even make sense? Because large language models, we typically have sentences. Let's say we have a sentence like, I am learning AI. And what happens in large language models is that we calculate attention between different words. So let's say we calculate attention between I and am and learning and AI. And then we basically encode this sentence, and then we can use it for different tasks like translation or even predicting the next word in the sequence. Now the question is that, how can we apply the same logic to a protein which looks very complicated, right? It has a three-dimensional structure. So how can we apply the same logic of a large language model to a three-dimensional structure, which is that of a protein? So essentially, we want to think about how can we mimic evolution by using large language models to predict new proteins based on a huge repository of sequences, structures, and functions. And this was something which I did not think really was possible, but recently I came across an update which kind of solves this problem. So this is a model which these authors have proposed. It is called ESM3, which is a frontier language model for biology. And what they have done here is that this frontier language model advances our ability to program and create with the code of life. And what they are essentially doing is that they are using large language models to generate new proteins. And I just want to show you one very cool application of this. There are these proteins which are called as green fluorescent proteins, or GFPs. When you see these beautiful jellyfishes having these amazing colors, it is because of the presence of these proteins which are imparting that color to that particular animal. And these proteins are rare to find. First of all, scientists have not been able to uh, bring about new proteins in these class of proteins in the laboratory. Whenever they are found in nature, that's when we know, okay, oh, this is a new protein. So it's hard to synthesize a new protein in this class of GFPs. And what these people have done is that they have generated a new green fluorescent protein which has only 58% similarity to the closest known fluorescent protein. And what does a low similarity mean? It means that we are predicting something which evolution has not yet reached. So we are predicting something which is far into the future, right? So if you look about if you look at this figure, now there is a lot of difference between this first shape and the last shape, right? And so the similarity is maybe 30%. So the lower the similarity, the longer is the gap for the mutations to happen. And 58% similarity is quite low, which means that we are kind of simulating way into the future, which is what these authors have actually achieved. And this is what their model ESM3 has done. And how it has been trained is that it has been trained on one of the highest throughput GPU clusters in the world. They have used a lot of data. It has been trained on billions of proteins, using which the algorithm has learned, and it is a multimodal kind of a model which can reason over structure, sequence, and as well as the function of the proteins. Yeah, so here is where it says language models generally operate over discrete units or tokens, and they cover three of the fundamental biological properties of proteins, which we discussed initially, which are the sequence, the structure, and the function. And this allows these ESM3 to be trained at generative generative capabilities. And it has a very simple objective. It uses masked language modeling, which is inspired by natural language modeling tasks. And at the heart of it, there is a transformer, as you might have guessed, because we are dealing with large language models. Now, the difference here is that it has something which is called as geometric attention. And why do we need geometric attention? We can't be using our normal attention like I discussed here. Geometric attention is where you look at the spatial positioning of, so let's say, uh, uh, what this means intuitively to me is that, let's say, we have a rod like this, and then we have another rod like this, and we have a third set of rod like this. So geometric attention means that spatially how close are these rods to each other. So what do I mean by that? So the angular difference between this and this is lesser than the angular difference between this and this, which means that the attention between, let's name it 1 and 2. So 1 and 2, the attention is more as compared to 1 and 3. So intuitively, this is what geometric attention means to me. So they have used geometric attention in this particular problem to represent their architecture. And this is just one of the examples of the masked modeling techniques, where they are generating a target protein to break down plastic wastes. And there are a lot of other applications also which they have proposed. If you look at this particular site, yeah, so they are imagining proteins to capture carbon, enzymes that break down plastic, then a tool for scientists to even imagine new medicines. So it's really amazing that we can actually define the function, and then based on the function, it will actually go ahead and it will give you the sequence and the structure of the protein itself. And then it really claims that we are simulating 500 million years of evolution, because we talked about this jellyfish, right, which has a very specific type of protein which is found in nature. And these people have simulated a protein which is also fluorescent, and it belongs to the same class, but it is not very similar to the existing proteins in this category, which means that we are definitely simulating a way ahead into the future. And then one another cool thing is that they have made this an open source model. If you go ahead and you click on this link, releasing the weights and the code, you can actually see the GitHub repo where they have uploaded the details of the model as well. And they say that, yeah, so you can use it for non-commercial purposes, basically non-profit purposes. But their idea is that they want to see what all people can create using this model, which they have recently developed. So just feel free to head over this website and explore this beautiful work. There is also this research paper which these authors have published, which explains the different things in quite some detail. And if you read the abstract, right, they start with that here we show that language models trained on tokens which are generated by evolution can act as evolutionary simulators to generate functional proteins that are far away from the known proteins. And if you look at the final structure which they generate, which is over here, so they have a section on generating a new fluorescent protein. And you can see here, this is the predicted structure which is shown in gray. and blue represents how different it is from the nearest protein. So there is a quite a lot of a difference it has from the nearest protein as well. And they have also outlined the methodology. So if you look at this first figure, you can see that they have used a masked iterative decoding. So instead of exposing the whole protein to the architecture, they expose it in an iterative fashion, and the transformer block uses a geometric attention. So anyone who is interested to read further into this domain, they can of course read this paper. It looks incredibly exciting. And why this is exciting is because it gives people the capability of generating proteins targeted towards a specific function. I really don't think this was possible 10 years back. If you would have asked people that, can we actually do this, people might have maybe laughed at you. But we are trying to mimic evolution now, which means we are really yeah, so it kind of means that we are understanding uh, the intricacies of the evolutionary process. Not we, the large language models, and then we are trying to synthesize proteins. So this paper looked very exciting to me, and I wanted to share with all of you. So just feel free to share in the comment section what do you guys think. And yeah, hopefully this was very interesting to all of you, and you head over and explore this in more detail. Thank you.